All right, welcome to part 3 of module 10 on cellular respiration. In part 3, we're going to be talking about fermentation, which is the anaerobic harvesting of energy, along with the connections between different metabolic pathways in the body. One term we haven't talked too much about is anaerobic versus aerobic. Anaerobic, as used here, means without oxygen. So fermentation is an anaerobic harvesting of energy. So it's the harvesting of energy without the presence of oxygen. The opposite of anaerobic is aerobic. Cellular respiration is an aerobic process where oxygen is required. Fermentation enables cells to produce ATP without oxygen. So fermentation is a way of harvesting energy that does not require oxygen. Under anaerobic conditions, muscle cells, yeasts, and certain bacteria can produce ATP by glycolysis. When oxygen is present, these types of organisms will produce ATP through cellular respiration, but when it's not present, they will produce ATP through glycolysis. This means they are facultative anaerobes. They can produce ATP in anaerobic conditions. There are some organisms that are truly anaerobic. Being exposed to oxygen would kill them. These types of organisms are typically bacteria that live in anaerobic ponds, so ponds that are so stagnant that there's no oxygen, or bacteria that live deep in a landfill and there's no oxygen that penetrates down there. All of those would be examples of obligate anaerobes because they cannot switch between aerobic and anaerobic respiration. But all of these listed here, they can switch between aerobic and anaerobic. In fermentation, NAD+ is recycled from NADH as pyruvate is reduced to either lactate, if we're talking about lactic acid fermentation, or alcohol and CO2 if we're talking about alcohol fermentation. Alcohol fermentation is how wine and beer are made. They're made using yeasts and they're forced into anaerobic environments. So like the wine barrels or the beer vats, those containers prevent oxygen from getting in because in those environments, the yeast will switch to fermentation or what they do in the absence of oxygen. We're going to look at lactic acid fermentation first. This is what happens in your muscle cells when they can't get enough oxygen. In order to still be able to produce ATP, they will resort to lactic acid fermentation. It's less efficient than cellular respiration, but they will still be able to produce ATP. It also was thought for a long time that lactic acid building up in your muscles was what caused muscle soreness after an intense workout. More recently, that's been proven to not be true. They now think it's related to little microtears in your actual muscles because lactic acid is broken down within an hour or two after formation. So this is the process of glycolysis. We see that glucose converts to the 2-pyruvate molecules exactly the same way as it did in cellular respiration, and it gives off 2 NADH and then 2 ADP are phosphorylated to become 2 ATP. We can see that this takes place exactly the same with or without oxygen. We don't need oxygen. Where the kicker is that NADH typically drops off its electrons at the electron transport chain, which ultimately what powers the electron transport chain is oxygen pulling those electrons down the energy staircase towards itself. Well, in anaerobic conditions, there's no oxygen, so there's no oxygen to pull the electrons down the electron transport chain. So NADH will not be able to drop its electrons off at the electron transport chain. However, in order for glycolysis to continue to occur, NADH needs to be recycled back into NAD+. So how microbes get around this in fermentation, in lactic acid fermentation specifically, the 2-pyruvate molecules are converted to 2 lactate molecules, and in that process, NADH can drop off its electrons and be recycled back into NAD+, which then can be used back into glycolysis. So lactic acid fermentation allows glycolysis to keep on continuing because NADH is being recycled into NAD+, by the conversion of 2-pyruvate into 2-lactate. So again, fermentation is a process which oxidizes NADH. Oxidation is loss, remember, so NADH needs to lose its electrons. It oxidizes NADH back into NAD+, which allows glycolysis to continue under anaerobic conditions of low or no oxygen, because NAD+ is needed to be an electron acceptor in glycolysis. Alcohol fermentation functions much the same as lactic acid fermentation. The only difference is that the final molecule in alcohol fermentation is ethanol. So glycolysis, again, functions the same. We end up with 2 NADH, and now we need somewhere for NADH to drop off its electrons because there's no oxygen. The electron transport chain isn't functioning. And that is where the conversion of 2-pyruvate to 2-ethanol will allow NADH to drop off its electrons and be recycled back into NAD+, which will then be available for the next round of glycolysis. In alcohol fermentation, 2-pyruvate is turned into 2-ethanol, and in that process, NADH can drop off its electrons and be oxidized to NAD+. And as a byproduct of alcohol fermentation, 2CO2 is also produced. CO2 is the reason for the bubbles in beer or champagne. It's a byproduct of the alcohol fermentation. And then ethanol here is a type of alcohol. One interesting thing about glycolysis is that it likely evolved early in the history of life on Earth, and we have a few lines of evidence for this. Glycolysis occurs in the cytosol of the cells of nearly all organisms, and it's thought to have evolved in ancient prokaryotes. This checkpoint question is going to kind of go through the reasons why. So the question says, list some of the characteristics of glycolysis that indicate that it is an ancient metabolic pathway. I'm going to just put the answer here and then talk through it. The fact that it occurs universally, functioning in both anaerobic fermentation and aerobic cellular respiration, is an indication that it evolved a long time ago. The Earth several billion years ago did not have oxygen available. When the first prokaryotes evolved, there was no oxygen, and so they would have had to rely on glycolysis, specifically anaerobic fermentation, until oxygen built up in the atmosphere. So the fact that it occurs universally and the fact that it does not require oxygen indicates that it came from a past when there was no oxygen, when those early prokaryotes were on Earth. Glycolysis also does not occur in a membrane-enclosed organelle. So we know that membrane-enclosed organelles evolved later on in the evolutionary tree. Prokaryotes do not have the membrane-enclosed organelles, so that suggests that glycolysis originally evolved during a time when membrane-enclosed organelles did not exist yet, which would be during the time of the early prokaryotes. So that's another line of evidence that it has been around a long time. And then the fact that it occurs universally also means that in the ancient lineages, those old ancient prokaryotes did glycolysis, and as different evolutionary branches of those original prokaryotes progressed, they retained that ability to do glycolysis. So all of those are evidence that glycolysis evolved pretty early on in the history of life of Earth. Only once membrane-enclosed organelles showed up did the whole process of cellular respiration come together. Until that point, glycolysis was the only way for cells to gain energy from the stored chemical energy of molecules. Now we're going to talk about connections between metabolic pathways. Cells use many kinds of organic molecules as fuel for cellular respiration. We've been talking about specifically glucose when we talked through cellular respiration, but that's not the only thing cells can use. So when we eat, we obtain most of our calories as carbohydrates, such as sucrose and other disaccharides, sugars, and starch, which is a polysaccharide, fats, and proteins. Cells can use all three of these types of molecules to make ATP in the process of cellular respiration. They just get inserted into the cycle at slightly different points. This image here shows you kind of where you can insert carbohydrates, fats, or proteins into cellular respiration. Carbohydrates can be broken down into simple sugars, which can be converted to glucose, which we know is the start of cellular respiration. So carbohydrates are easy. They come in right at the very beginning. Fats can be broken up into their component glycerol head and fatty acid tails. The glycerol can then be converted to G3P, which is one of the intermediate molecules in glycolysis. And then the fatty acids can be converted into acetyl-CoA, which is that product of pyruvate oxidation. So they get inserted into the cellular respiration cycle at these points. Finally, proteins can be broken up into their component amino acids, which can then be transformed into pyruvate, which is the product of glycolysis and the reactant in pyruvate oxidation. Amino acids can help form acetyl-CoA, acetyl-coenzyme A, the product of pyruvate oxidation and the reactant in the Krebs cycle, or the amino acids can just be inserted directly into the citric acid cycle. So all of the different things you eat can be inserted at different points, and that's kind of what this figure goes on to show. A checkpoint question here: can a human survive on a diet consisting primarily of fats and proteins and almost no sugar? Would cellular respiration still be able to occur? Take a second and think about this, and then we'll come back together. So if you said yes, you are correct. Breakdown products from fats and proteins can still enter the cellular respiration pathway through glycolysis and the citric acid cycle to make ATP, which is what we just talked about in this image. You don't have to be consuming carbohydrates to enter the cycle as glucose. Fats and proteins can be broken down to enter the cycle at different points as well. The last topic we're going to talk about today is that organic molecules from food provide raw materials for biosynthesis as well. So not only can biomolecules be broken down and enter cellular respiration at any point, your body can also take out biomolecules or components of biomolecules from the cellular respiration process at any time. Cells use intermediates from cellular respiration and ATP for biosynthesis of other organic molecules. This image shows that over here. So we can take, this is kind of it's going in reverse. So this is how we would get glucose. You would go from pyruvate to G3P to glucose. You also, if you have G3P and your body needs more fats, you can take the glycerol out of the G3P molecule, combine it with fatty acids from the acetyl-CoA molecule to make fats. Amino acids can be pulled out of the citric acid cycle. They can be pulled from acetyl-CoA or pulled from pyruvate to form proteins. And then again, sugars can be pulled from glucose to form carbohydrates. And these proteins, fats, and carbohydrates are what make up cells, tissues, and organisms. So this is just the reverse image from what we just looked at. Any of these intermediates can also be pulled out of cellular respiration if needed and converted into biomolecules. Whether or not this occurs is often regulated by feedback inhibition. So if you already have enough fatty acids or enough fats in your body, feedback inhibition is going to prevent any of these from being taken out of cellular respiration. But if you do need more lipids, more fats, then your body will start removing fatty acids from acetyl-CoA and G3P from glycerol to synthesize those fats. Our final checkpoint question: explain how someone can gain weight and store fat even when on a low-fat diet. If we take a look back at this figure, you don't have to be eating fat in order for your body to make fat. We see here that even glucose, as it's going through cellular respiration and being converted to G3P and then pyruvate and then acetyl-CoA, at any point in that process, the intermediates can be taken out to make fat. So even if you're not actually consuming fats, you're only consuming glucose, through these intermediate products of cellular respiration, your body can still synthesize fats. So if caloric intake is excessive, body cells use metabolic pathways to convert the excess to fat. The glycerol and fatty acids of fats are made from G3P and acetyl-CoA respectively, which are both produced from the oxidation of carbohydrates, specifically produced as glucose moves through the cellular respiration cycle. That's the end of this lecture. That was a pretty short video. I'll see you guys again in Mod 11.